Hello. Hi everybody, welcome to the Mechy colloquium. Are we recording? Yeah, okay, great. So I'm delighted to welcome Professor Neelesh Patankar. Both delighted and honored that he's here. And Professor Patankar is a professor of mechanical engineering at Northwestern University. I've known him for many years, and it's been a great privilege to collaborate with him, but also learn from him on many topics. He has done seminal work in computational methods for aquatic locomotion, among others, fluid structure interactions, and he's been doing work on mechanophysiology, which is a topic today, on surface engineering, electrokinetics, and so on, so forth. Many interests and many topics, I hope you'll have a lot of things you'll take away from his talk today. He has also won a number of awards, including NSF Career Award, to being an APS Fellow, to more recently at the microflow and interfacial engineering seminars, where he has been given the highest honor. This has been a society that formed over maybe the last decade or so. And he also serves on many editorial boards, including ASME Journal of Fluid Engineering and others. And yeah, I've always found his work, back when I was working at GE, we found a lot of his work to be very foundational to some of the things we have done. So I'm really delighted that he's here, and with that, I welcome Neelesh to talk about mechanophysiology, and looking forward to learning about this topic.
Thank you, Kripa. All right, so I have to turn this on. Okay, so I'm good, right? Yeah, okay. Yeah, thanks, Kripa. Yeah, and just for the students' sake, I will be here actually until end of March. Thanks to him and Department of Mechanical Engineering here. I have been on this 6-month stint here. I've been here for a few months now already. I've been enjoying some of the students I've talked to are here in the audience, so hopefully this will lead to some nice collaborations going forward. First off, I just want to start off acknowledging, because that is the last thing that gets missed many times, NSF, NIH, and most important, I want to give a shout out to my great collaborators, John Pandolfino and Peter Karilas. They are physicians. Everywhere you see doctor, they are physicians. The PhDs don't get a doctor in front of their name here. So, because physicians say that those are not real doctors, right? So anyway, so we work on many different organ systems, and a lot of the work today I'll discuss is gastroenterology and cardiology in the context of a bigger framework, which I will discuss today. I want to give a shout out to my students. Walter is the one who got me started in this. Shashank came up with the first diagnostic metric for esophagus based on our work. Saurav developed this concept of virtual disease landscape. Guy Elisha was the first one to do a neuromechanics model for esophagus ever. And then Tom Zhao did some aortic aneurysm great work. So just a shout out to them. All right, so what do we mean by mechanophysiology? It's a narrow term in some sense. It's physiology is function of organs, and mechano is essentially saying mechanics of organ physiology. So I'm focusing on organs where mechanics is the key determinant of its function. But it need not be so. In fact, you will see that today itself, it could be extended to other physics areas, and one is neuromechanics. Okay, so you have these electrical impulses also accounted for. So, and this particular project, which like I said, I started working 10 years. It has become a passion project for me for a variety of reasons, personal and so on. All of us know somebody who has had some medical issues, and it really becomes a passion thing in terms of seeing how you can impact this area from our vantage point, okay? So again, these are the different organs, and just a little bit of background as to how I got into this. So like Kripa said, I have several different lives, and then people in one field don't know what I'm doing in the other one, so just orienting. So I do a lot of surface engineering. I'll probably give a talk in one of his lectures later on. But I also have my native space in fluid structure interaction algorithms. So a lot of applied math work. Vim and IV overlap in that area. and I have applied those to different problems, aquatic locomotion being one of those. Okay, and then 10 years ago I started working in mechanophysiology. And just to give you a visual as to the kind of stuff we started doing, here is a simulation of bolus transport, liquid bolus passing through a esophagus, which is just the straight part here. There is no stomach here. So I'll just lead you through it so we are oriented and we know what we are going to be looking for. So this black mesh there is the muscle layer, okay? And that muscle layer is a matrix fiber structure, okay, in this modeling. And that two types of fibers are there, concentric with each other. Here I'm showing it separate, circular fiber and longitudinal fibers, okay? This blue mesh is what is called mucosal submucosal layer, and you can see it kind of slides. It's compliant as the bolus passes. It's just like your skin. So in fact, if you take a esophagectomy sample, you will actually see that you can kind of pull and push it in and out. Okay, so that is how it is. All right, so we do these kinds of simulations. You get velocity field, pressure fields, and so on and so forth. Just to make a note, of course, we also have a stomach model connected to this, and we can study reflux and stuff like that. All right. So using these kinds of tools, we have looked into traditional biomechanics approach. You look at all the different fundamental insights you can get. And some of the things we have done is, okay, what is the role of circular versus longitudinal muscle in this? What is the role of this submucosal layer in this bolus transport, and so on. So just to demonstrate how we have used this for fundamental investigation, I'll give you one short story. Okay, that is not the point of this talk actually, but just to set the context. So there is an example, and this gives me a chance to introduce one device to you, because that is going to be part of our story today, and that is called esophageal manometry. Okay, so if you have some esophageal problems, I hope you don't, you go to the doctor and they will put this catheter through your nose and you will be asked to swallow something. Okay, and the catheter has pressure sensors all along the length over here, from the upper esophageal sphincter, which is the sphincter at the top, to the lower esophageal sphincter, which is the one that goes into the stomach. The food goes into the stomach through it. And it will get a pressure reading. And this is the kind of map which is called esophageal pressure topography, or EPT, that is made mouth to stomach. So that is this vertical axis. Horizontal axis is time. Okay. So if you just think about it for a minute without looking there, if something is squeezing and pushing your bolus down, so squeezing place is where you will see high pressure, a red color, okay, over here. And you should see a uniform red band going down diagonally. Okay, you don't. All right, what you end up seeing is this transition zone over here, and that has been a question of discussion in this field. And there is a resolution for that, and that is the upper part of the esophagus, or proximal, versus the lower or the distal part. They are innervated differently. Okay, and as this contraction wave or the neural activation goes from upper to lower part, this handshake process creates this kind of a bump, which creates this transition zone. That is one hypothesis, okay, and people have evidence for that. But as we were trying to develop, again, I'm trying to give you an example of how we have used our model, mechanics model. As we tried to develop these models, we wanted to know what is the fiber architecture that we should use, right? And we saw that actually from some literature that the fiber architecture transitions from the proximal to the distal part. So it is more like this and becomes more like that as you go from top to bottom. And our hypothesis was actually, even if there was no innervation jump, right, you would still get that kind of a behavior because of the fiber architecture change. That was our hypothesis, and we could, that is a good part of simulation. You could do simulations, switch on and off different mechanisms and check what happens, and lo and behold, we actually are able to reproduce the kind of pressure topography that is seen in real situations. Okay, so this is again a demonstration to just show, okay, this was a normal case. We were able to also reproduce several different scenarios where experimental EPT. This is obviously a patient one. This left side is a normal subject, and we are able to reproduce this. Okay, so right now I'm just trying to convey that this is the kind of stuff that we were doing. All right. So what about it? Why is it important? So let us first see how the doctors use this EPT that has been collected from manometry. Okay, and then we will see how we could use our mechanics stuff. Okay, and that is going to be, I'm setting up the whole idea that we are trying to do in our work, right? So So when they take this manometry, it goes into some kind of an algorithm that they have based on their empirical knowledge and their thinking. And my collaborators, they are known for this Chicago classification. Here I have picture version 3, latest version is version 4. Currently, we are writing a proposal for version 5. Okay, so it keeps getting updated. So this goes into this, and then it gives a diagnosis. Okay, so that is the basic idea. What are we doing? What we are doing is, of course, we are trying to unravel what caused that thing, okay, that particular pressure topography, like the example that I just showed you. So this is great to have some insight fundamentally into this. And this is the kind of biomechanics work that has traditionally been done over the past 50, 70 years, okay? So, and computational work more so in more recent years. So now the question is, how can this be actually used to influence that? That part was not clear, because what happened in the end was we were told that, okay, fine, you are creating this fundamental understanding, but how will it help us classify diseases better? Because this is what is happening here, disease classification, okay? So what you need for that is you need diagnostic metrics, okay? Some mechanics-based metrics like blood pressure 120/80, for example, right? So above, below, high, low blood pressure. So some kind of a metric which is tested, and some kind of a disease classification scheme that goes there. And now there are so many different kinds of disease, so now it is suddenly disorienting for us that, okay, so what is the organizing principle by which I can systematically do this classification, which is mechanics based, that can be used by clinicians, okay? So that was one of the questions, and this is a broad question, nothing to do just with esophagus, okay? This is a broader question that we realized. Second thing we realized, that whatever mechanics based parameters I put in there and come up with metrics, what is going to happen is the moment you put d by dx or integral, it becomes a problem. Okay? And I'm not saying this as a criticism. I'm going to, in fact, point out why this is so very soon in a few more slides. But what that means is whatever metric calculation process you have to integrate it with your tool. Okay? Otherwise, it is not going to be used. Okay? So those are the two lessons we have learned, and in that sense, we decided to formulate some generalized framework so that we can systematically dig in and be useful for diagnostic for these physicians of different organ systems, where mechanics is one of the key determinants. Okay, so that is the story. So that is where I'm trying to go. Clinical medicine, okay, engineering medicine is a broad term, but clinical medicine in the context of diagnostics. All right. So where is that? Okay. So before I go there, okay, so I'm trying to help develop this disease classification. I want to indulge a little bit and make a slight detour and tell you a little bit of historical story. Okay, so that way we will know that currently the way disease classification is done or the way medical education is happening, how the doctors think, we will have some context in which now we can try to make a difference to them. Okay, so that is how at least I started thinking. And for this, you have to go back in 19th century, 1800s, and at that time, if you look up some history, one thing is that a lot of therapeutic practice was based on understanding of function. Okay, so they were focusing obviously on what you see, okay, the function of that organ. So that is physiology, okay, anatomy, physiology. So we'll remember that. And by 1870s, germ theory happened and drugs started being used, and therefore the importance of biochemistry started arising. Okay, so these two things happened around that time. And however, the medical education apparently at that time was a little less organized. Okay, so in early 1900s, the American Medical Association, they sought a survey from the Carnegie Foundation, okay, asking that, okay, what should be done to organize the medical education in United States. And that task fell upon somebody called Abraham Flexner, and he came up with a report which came out in 1910, okay? And this has been considered to be one of the most influential reports for medical education and training all over, not just in the United States, okay? Now this report does have certain aspects which are racist, okay? But right now I'm not focusing on that, okay? A lot has been written about that. but let us see what are the things that are still there today. Okay, so one of the, and I'm not saying these are bad things, these ones that I'm listing here. I'm just trying to understand what the context, right? So what was proposed was physiology and biochemistry, and this becomes obvious from the previous slide that I had, why that was so. And because of which, there was a lot of focus on physics, chemistry, and biology, okay? And in fact, if you go in countries like India, in US, because the undergrad degree can be anything, engineers do become physicians. That does not happen in other countries, okay? So if you go to India, after 10th grade, meaning 11th, 12th, people would not have math. Remember, look, math is missing there, okay? And 11th and 12th is where calculus-based mathematics was learned, okay? So we are getting physicians who have not learned calculus-based physics. They know physics, it is listed there, but they know physics like half GT square algebraically, okay? Not through the language of calculus, all right? Of course, statistics has been there. Okay, so just know this context, and this explains when I told you that you start putting integrals and derivatives. Yeah, sure. In some places, like the place that we are here at, or maybe at Northwestern, you will have physicians who are trained to do that. But in general, they are not. All right. So that is why I'm doing this story. And then, of course, biomedical engineering has been around for a long time, 50, 70 years now, and a great work has been done. But somehow I feel, okay, that in clinical practice it has not been integrated yet, and I'm trying to suggest what we could do to change that, okay? So, and this is again the restatement of what I just said. What is the impact of bioengineering? Of course, a lot on tools and devices side and a lot of biotechnology side, but in the clinician's, physician's room, right, where they actually meet you, right, there when they do diagnosis, how much of it is based on, for the physical function of the organ, not based on pathology test, okay? So that is a different biochemistry angle to disease diagnostics, okay? So this is where we want to make an impact, okay? So that is the point. And currently, how is this disease classification being done? In fact, you can go to this website organized by World Health Organization, and you will find this is an encyclopedia of all the different diseases for different organs, okay? And you will see they are based on symptoms, okay? And whatever has been learned so far. But how does it fit? How are these different diseases for a particular organ related to each other? That part is not always clear, all right? And then for particular organ system, there are these Chicago classification schemes, version 4 in this case, similar to the one that I had shown you before. And this is where we want to insert our mechanics-based thing, okay? So this is the context that I am trying to lay out, all right? So let's now see how we can insert it into this system, all right? So what we decided was, okay, so let us organize yourself by thinking from our native space, which is what kind of organizing principles in physics or engineering can we use to start, getting a hold on this problem, right? And for me, the simplest organizing principle was pi theorem, okay, Buckingham's pi theorem, okay? So this is, what do I mean by that? Okay, so here is an analogy. Imagine there is a sphere, the one on the left, analogous to an organ, which is the esophagus in this case, okay? And you are stimulating something, setting up a problem, which is material properties, boundary conditions, and so on and so forth. So these are all the things that you would put when you have, a system of equations you are trying to solve it, you give boundary conditions and material parameters and so on. So these are all the input variables which will cause some output, which in this case, let us say we are interested in drag, analogous to how well you swallow, okay? So that is the output function. So this output function will be function of all these input variables. And of course, these are functions of these physical variables, but that is not the efficient way to think about it, because if you use dimensional similarity, we know that actually you can reduce it into CD versus RE, okay? And there is that one pattern, CD in this case, that graph, which explains everything. All right? So we can imagine that the same thing can happen now for understanding this disease classification, right? So whatever is the behavior or function of that organ in the mechanical parameter space, I can organize that potentially, right, in terms of non-dimensional parameters. Here I'm showing 2. And let us imagine that the third axis is probably the function, whatever it means to say swallow. Instead of having the third axis, I'm putting colors, okay? So these different colors are the different type levels of function of swallowing as a function of those non-dimensional parameters. So I have this mapping, And now, if I am going to classify diseases, right, I have these fundamental non-dimensional parameters which are like my basis vectors. And I'm talking linear algebra because actually when we do the landscape, that is a lot of linear algebra. So thinking about this diseases like disease basis vectors actually helps. Okay, so that is why I'm introducing like that. So that is the basic idea. Okay, so can we do this? And this will happen. The disease or functions will cluster or separate away if our hypothesis is correct that the function of that organ does primarily depend on mechanical parameters. Okay, so those are the organs that we are talking about. And in fact, here is a list of non-dimensional parameters that we have for the swallow function. We have done all that, and one could come up with different insights based on that. All right, so said another way, this is your system. This is some reduced order model for it. And we know that given some input, which is what we call stimulus, right? If you have no stimulus, nothing is going to happen. You need some boundary condition or whatever which drives that system, and you get output. So when diagnosis happens in doctor's office, it happens based on an understanding of this. And they observe this, and based on that, they are trying to deduce what might have caused it. Okay? And most of that thinking obviously happens in natural language, okay, which means English or whatever your language is, or some understanding of algebraic physics. And what we are trying to do is, okay, let us try to deduce it based on, like bioengineers have been trying to do for 50 years, right? That, let us try to deduce it based on our calculus. And of course, this is a ill-posed problem, doing this inverse problem. But using guidance from physicians and putting some reasonable constraints, you can come up with good insights, as we will see. But this inverse problem, having it completely in this space, is an aspirational goal, not always possible. So you will see when I show results, it will be a mixture of, for that landscape that I told you, it will be a mixture of input and output variables in a judicious way. All right. So if I, in principle, know these input variables, these are my levers, which is actually deciding how the function of that organ is changing. So I can have that full map for it, okay, in principle. And that will help me define different disease basis states, right? Like those basis vectors, there are what are the principal types of diseases that are there. And that will give me the mechanistic origin of it. And depending on boundaries between different types of functions, I could have thresholds which can be my physiomarkers for diagnosis or classification. Okay, so that is the basic idea. Not only that, we could potentially map each of those states in this mechanical space to the underlying genetic profile, okay, and try to understand what is it at the molecular level that is causing these mechanical dysfunctions. Okay, so that is the overall picture. All right, so let us keep going here. This is that space. So what happens is that these are my disease basis vectors. In principle, this is what we'll call virtual disease landscape. This is the parameter space that defines the mechanical health of that organ. Okay, in that, different functions or different disease groups will cluster in different parts based on these different colors over here. And a particular subject or a particular patient will be a dot on this mechanical parameter space. Okay? And you can track the progression of disease on this for a particular patient over time. Or you can track how that patient changed based on any treatment that has been done. Rather, you could eventually plan what kind of treatment should be done so that the patient moves in the right direction. Okay? And I'm insisting on the word landscape here because think about it like this, that you have some function plot which is like a hill and a cliff, right? So many times your functions are looking good. I'm going to give you an example based on aortic aneurysm later on. Your parameters look good, but you're very close to the cliff. Okay, so they deviate a little bit and suddenly your function goes down and something catastrophic can happen. Okay, so if you have this landscape, you know where the border is. It helps you predict whether you are in a precarious position or not. Okay, so that is the basic idea. All right, so let us see an example of this virtual disease landscape in action. Okay, so I will now introduce another device over here. Okay. And that device is essentially called FLIP, as you can see up there. Okay? So the difference between this device and the previous one was it is a saline bag, as you can see here. Okay? It is basically filled at different levels. Okay, so they will change the volume to which it is filled, and the patient is sleeping. Okay, they have been anesthetized, and the bag will be inflated at different level, which stretches the esophagus. Okay, and inside there are sensors which senses the local cross section area in principle. All right, so and this is that picture over here. So you are seeing a video of how that cross section changes with time. All right, and what ends up happening is your esophagus gets stretched, and there are, this is the hypothesis that the stretch receptors, they activate the cells there and create contraction patterns which go top down. All right, and that is what you are seeing. But not only that, it doesn't do one contraction and stop. And remember, these are not conscious swallows because the patient is sleeping. Okay, they repeat. So these are called repetitive antegrade contractions. Okay, so why is this important? Because imagine that you are swallowing and your food doesn't pass through and it stays in the esophagus, right? So it stretches locally the esophagus, and that creates these secondary contractions. So therefore, this kind of thing is called secondary peristalsis rather than a swallow-induced one, which is called primary peristalsis, which manometry checks. Okay? So this secondary peristalsis is a mechanism to kind of try to keep pushing the food down that has been stuck. Okay? So that is the basic idea behind it. All right. So this flip device essentially checks that. All right. So now this happens, and correspondingly there is a pressure sensor right here, and that pressure reading has this fluctuational component because this contraction wave goes down, relaxes, goes down, relaxes in a repetitive fashion. Okay, so that is the basic idea. Now, before we go further, like in the bigger context, one has to be careful. Okay, so I'll say a few things so that it helps develop context. So imagine your parameter space is two-dimensional. Okay, for the disease landscape. Now you have a device that is able to probe this axis, but not that one. So it will be blind to pathologies found because of this axis variation. Okay? So when you have a particular device and you try to develop a virtual disease landscape, you may not be able to capture all the diseases. So we have to be mindful of that. And eventually, you might need a minimum set of devices that explores the entire space that gives you the real mechanical health of that organ. Okay? That is at least in principle. So now we will see how well this works in terms of capturing different things. All right. Okay, so the way we proceed in terms of developing a virtual disease landscape based on this device is now look at the pressure sensor, and you will see that I told you earlier, as shown by this red plot, that bags are inflated at different levels. Okay, so they will add certain amount of fluid, wait, take data, then increase it, and so on. So as the volume of the bag is increased, you're stretching the esophagus more and more, and therefore the pressure basically increases more and more, and the bottom end is the most relaxed. Okay, that is like the uncontracted state of the esophagus. Okay, so you get the passive properties approximately of that organ. Okay, and that is basically this line that I'm showing here, so which you can plot as pressure versus area, and using a linear tube law, which has been established for esophagus in the past, you get this K over A naught, where A naught is the reference area of the relaxed esophagus at a particular location versus this A, okay, is the actual area. So you get those passive properties. So that is step one. Okay, so we are getting those mechanical parameters. Now you go back to this, and you have area versus time and location, okay, X and T. You put that in this reduced order model that we have done here. You could have higher order models, mass conservation, momentum conservation, and tube law. The only thing I will note is, this tube law has this theta here. Because of contractions, the reference length changes. Instead of being A naught, reference length is A naught times theta. So if theta is less than 1, you have contraction, theta greater than 1, you are relaxing that muscle. Okay? So the question is, given this observation, what is this contraction pattern? Because that is the important one. That tells you the motility of that esophagus. Okay? So that is what it is. So you do this problem and you get this graph. Okay? Length versus time, this is the color graph of this theta, less than 1 contraction and greater than 1 relaxation. All right, so now these patterns are important, and this pattern will repeat. This is just for one contraction, by the way. Okay, so these patterns repeat. So what we need to do is, we are told that, okay, don't make that through any reduced order model. because those patterns are important. So we decided, okay, this is 256 pixels here. So we will try to reduce the dimensionality, and there are various ways to do dimensionality reduction. We decided to go with AI technique called variational autoencoder, which has this latent space which is reduced dimensional 24 dimensions over here. Okay, the reason we use that is it did a good job. Okay, there are other methods as well, by the way, and it has a generative component, so that even if you don't have a data for a certain location, you can predict how something will behave if you pick some data point that is random. Okay. All right, so that is for this theta, for this contraction pattern or the motility behavior. And then on top of that, there are few more parameters, forget about this, which includes that K over A naught, that stiffness and all those things that are additional parameters over here, okay, so which I have not listed. So this together is that parameter space that really defines my virtual disease landscape, okay. Now what I'm going to do is, I'm going to do in 2 steps. First, we will look at this one, okay, the motility one, and we will observe it in a reduced order space just for our visualization in 3 dimensions. Actually, it is 24 dimensions. And we will take another example where we have this contractility behavior together with the stiffness behavior and try to see how that helps diagnose diseases, whether they cluster or not. Okay? So now what we have done is different patient groups. We calculate this theta. It goes and sits as a point in this latent space. Okay? So it's all put there. Data is there. Now, this is unsupervised learning, right? So we are not telling them any disease type when we send in the data and create this cloud of points. Okay? And then we say, okay, now let us color those points. Okay, based on the classification that the doctors had for this, based on this device. So there are these 4 major groups, and we decided to color those. And now, if our hypothesis of virtual disease landscape is correct, meaning that in the mechanical space different diseases will separate, then automatically you should see those colors in separate areas in that regime. Okay, and that is exactly what happens over here. Okay, so and now you are in business, because now you have kind of boundaries, or you could set probabilities, and those become your thresholds for differentiating different disease groups and do disease classification. Okay, so one thing you will notice here, we did not create the definition of good function based on some mechanics based assessment. We actually followed the doctor's judgment and their disease classification and told them how the diseases they have classified we can separate based on mechanics based parameters. That is very important, okay, because now that it is working with the doctors and using their judgment. That is very important. Okay, so I'm going to skip the next one. So this is the slide which is a validation of showing that there was a patient in this case right here in this red area, which is the bad region, and some procedure was done called POEM. Okay, it is about tearing some muscle from inside of the esophagus, so it loosens up and swallowing improves. So the patient reports that the function has improved. Okay, so this is the reality. Now, if our virtual disease landscape is correct, then the place before and after should be such that after should be in the better area, right? Because the patient is actually saying that they are feeling better. Okay, that is kind of a test of our thing, and later on we can be predictive about it. But first we have to do validation. So this is in fact showing that that is true. Okay, so it is going in the right direction, and in fact it is borderline. As you can see here, this is not a fully normal behavior. It is borderline behavior. Okay, so this is correct. So, another thing that doctors like to do is that they will see whether the patient is stable or not. Okay, and what we are doing here, okay, so patient stable, 1st year, 4th year, 7th year, their dots are in that same borderline neighborhood area for this particular patient. So they are thinking based on their current judgment that this patient is stable. We are showing that it is in fact the case in our virtual disease landscape, but we can quantify it. That is the main point. Okay, so quantification is the key thing, and then later on we can be predictive, hopefully. So these are all validation cases I'm showing to make a case. Okay, finally, finding some additional metrics for disease classification. Okay, so here is one. So you see this blue patch is normal group. Okay, and when you are swallowing normally, your esophagus opens easily, and the amount of work that you do is certain amount, it turns out to be around 100 millijoules. Okay? So this is the first time somebody has calculated work done by the esophagus. All right? So now what we do is for every group, we measure the actual work done by the esophagus in that flip. All right? And this is that cluster. Okay? Darker color is more, enough work is been done. And lighter color means the esophagus, the sphincter is not opening, so enough work is not done, the food is not passing through. All right? So as you can see, the work done is clustered around this blue group. And in fact, you can create thresholds of how much is the normal work to define a normal subject, okay, in terms of the work done. So work done becomes a metric that you can use to identify which subject is normal and which are not. Okay? So this is an example of coming up with a physiomarker, okay, or a metric to do diagnosis. Okay? So, all right. So I'm going to skip that. So this is so far, like I've tried to establish that, okay, this principle of virtual disease landscape is useful, okay, and at least can be demonstrated for the case of the esophagus. And I'm going to give you one quick case study, okay, of use of this, where I also include the stiffness of the esophagus as one of the parameters. Okay, so this EOE, or eosinophilic esophagitis, is a fairly recent disease that has been categorized. And as you can see from Mayo Clinic, there is still research going on as to what is the best way to identify it. All right. And the symptoms are, difficulty swallowing, food stuck and pain and so on. And if you do endoscopy at later stages, what you will end up seeing is this kind of a structure. And there is a greater eosinophil count that you see in the blood work. Okay, so that is what it is. But in the early stages, you may not have all these things showing. Okay. So what is done? Okay, so you do flip. Okay. And what has been observed that these EOE cases, they seem to have these 6 types of flip patterns. Okay? So now these are plots of the cross-section area, length versus time. Okay? So this color plot is not pressure, this is cross-section area in the flip. Okay? And just if you see visually, there are 6 patterns. Are these 6 phenotypes, as one is calling it, or is there some consistent pattern underneath it? So we applied our VDL concept to it, okay? And the questions we were trying to answer are these different subtypes? What is basically the quantitative diagnostic scheme for this, for EOE and so on, okay? So that is what we were trying to do. So again, we did the same thing. This time I'm showing you 2 axes. Instead of all axis being based on that theta contraction pattern, I have one axis based on theta. Okay, this is using something called LDA. So this is contraction axis, and this is the stiffness axis. Okay, and those are all the 6 groups out there, and it might look they are intermingled. But when one tries to find these thresholds, in medicine, what is normally done is you find the middle 50%. Okay, so here is the 6 groups of K over A naught values, middle 50% of each group. And you can see there is a clear categorization. There is a normal K over A naught range over here. Okay, blues are the normals. And then there is increasing K over A naught. Okay, so clearly there is a gradation, and there is a threshold for K over A naught between normal and getting stiffer. And if you see the contraction strength, which is that CR, okay, there are 3 categories. Okay. So there are 6 groups, but they combine 2 by 2. I'll show you how. And there are 3 categories, and this is the normal group here. Okay. So again, there also you can identify thresholds. I will draw those thresholds. And just to make it clear, so one good and one bad K over A naught is clustering into one CR. Okay. And those are those 2, and those are those 2. Okay. So that is how they naturally go. In fact, this is an important result, because one of the things that was there is if it gets stiffer, the question was the contraction changes with it. And in fact, this is showing that that is not necessary. Okay? So these are 2 independent things. K over A naught changes and contraction strength changes, and they need not always be correlated. Okay? That is what this is already showing. Okay? All right, so having said that, you can identify these thresholds, and without disagreeing with the basic classification made by physicians, we are trying to find thresholds that agree with it, and now we can recolor this to be consistent with that statistic, okay? Because there is a lot of gray area in terms of classification, okay, when physicians do it. And this is the recolored plot right here, and you have these 6 groups, okay, over there. And in fact, you could also redraw this plot. Instead of the contraction strength, you can plot it in terms of some other variable, which is the displacement of the volume as the contraction goes down. So that is another variable you could use as a proxy for this contraction strength over here. Okay, so we just did that because the physicians wanted it. Okay, so you stare at this and now you start thinking, okay, so are these 6 different disease types, or are they connected as one follows the other as a disease progression? Okay, so one could hypothesize that actually there are possibly, okay, we haven't proven this, these 2 phenotypes of progression, okay, that the contraction strength goes bad and there is other where stiffens and contraction strength goes bad. Okay, so that is something that one could study. All right. So all this happens and you are still thinking that, okay, so you know this changes in the mechanical space, but what is causing it at a molecular level? Okay, so what you could do is potentially, and that is the part we haven't done yet, but that is something that is in the works, that for each of these colored groups, you can basically get cells potentially, do transcriptomics on that, and try to understand any genetic or epigenomic changes that might have happened which have progressed over time. Okay? So you can have a map of these physiomarkers, and behind it is a layer of these transcriptomic data, okay, which tells you how the molecular picture. So you have this gene to function picture that can happen. And the preliminary work our collaborators have already done. For example, it is shown over here. It is a basic graph, but this group here, these first three bands, these are all healthy groups, and these next three bands these are all EOE, all of them together, not separate colors. Okay, what we want to do is for separate colors eventually. And what you can see is, and each of these spectral looking lines, these are gene expression lines. Okay, so what you can see, okay, is that essentially if you see this disease group, look at this band pattern, it's not the same as the normal pattern. So clearly the gene expression behavior has changed. Okay, and one could then track how it has changed through disease progression if you track it for different groups. Okay, so that is the big picture in trying to go all the way from physiology to genetics. Okay. All right. So how much time do I have? Okay. So I can tell you a couple of quick stories. Okay. So so in all of this, you can see that one of the things you noticed that okay, theta, right? That contraction pattern. Why does it change, right? I took it as a given right now, right? It has become bad, and then based on that, oh, this is how the function is, okay? So given theta, this is the function. That is what I talked about. But why did theta become what it was that caused bad function, okay? If you want to understand that, you need to do this neuromechanics, okay? So I'm going to tell you a very quick story on that. Okay, so there is in fact a basic question. So this is an additional layer that will go underneath that theta, okay, in our mechanical model. Okay, that is how you build this model. So going back to this, so if you plot this contraction pattern as a color plot, you can imagine these are going to look like that. Okay, red bands going diagonal, and this is exactly the repetitive antegrade contraction that you have. Now I told you earlier that the basic understanding at this point is that the stretch receptors, you stretch the bag, right? You stretch the esophagus, and that triggers excitation and that causes contractions. Okay, so that is what I told you. But actually, if you see, there are still questions. For example, there has been a recent high-profile commentary on this, questioning whether that is really true in case of flip. Okay. And there is no neural circuitry that explains that repetitive pattern. Okay? So, and this is important because remember, minimum set of devices, right? If manometer is picking primary peristalsis and flip is supposed to pick secondary peristalsis behavior, is flip really picking secondary peristalsis behavior or not? That is the question. Okay? So that is really fundamental, and we try to interrogate that, and that is what I'm saying here. Okay? So these are the two things we decided to probe. Okay? Is there a neuromechanical model that can explain this? And hopefully through some animated slides, I'll be able to convince you that there is an answer to this. Okay? All right. So don't bother about the details of this, just the key points I will mention. So E means excitatory cell. So imagine your esophagus top down. I is the indicator which lower number to higher number it goes top to down. Okay? So when you are at EI, at some level in your esophagus, there are certain number of cells that is going to be excited. So the proportion of cells that are excited is E. Proportion of cells that are inhibited, which causes relaxation, are I. Okay? So E causes contraction, I causes relaxation. Okay? So now what we are saying is stretch receptors, which are I, stretching, mechanical stretching, is causing local cells to excite, and local plus all the way downstream cells to relax. Okay? And this is the thing that we have not come up in thin air. This is a proposition based on literature. Okay? So this is an empirically built model, okay, of the neural circuitry. All right? And the question is, will this produce that emergent behavior of repetitive anterior contraction and actually reverse contractions, which I didn't focus on in the previous slide, but I'll come to it. All right? And then the second point I want to mention is this excitatory cell, they activate this theta. Okay? So that is what the mechanics part is. So this bottom part is mechanics. So let's get to the equations. It's hopefully not going to be too difficult. So these are the equations for excitatory and inhibitory cells, and those circled ones are the ones taking input from the mechanics part, which is the stretch receptors. Okay? And these are some detailed equations you can ignore. On the other side, there are mechanics equation, mass, momentum, and tube law, where the theta is the one that is being activated based on excitation, as you can see in the model here. So this is input to mechanics from neural, and these are input to neural from mechanics. Okay? So that is the whole idea. And the cascade of things is area is caused by theta, which is caused by E. So let us focus only on this E right now to get the pattern understanding. Okay? So let us, for pedagogical reasons, keep it simple. All right. So now I have these different cells, okay, at different levels, and they are connected one way, okay, in the sense that the upper ones affect the ones lower down, not the other way around. Okay? So that is an important empirical information we have used. Okay? So now if that is the case, for a minute, let us imagine that they are all decoupled across the length. Okay? So that is not there. Okay? Let us pretend for a minute that there are no stretch receptor activation either. Okay? So I'm trying to set it up, and now we are trying to find a steady state. So this E dot and I dot is 0, and the corresponding steady state equation for this is this curve, and for that is the other curve. And so if you want a collective steady state solution, it will be the intersection of these two lines, which is that, which means nothing is happening. Okay? Obviously, if there are no stretch receptors, nothing should happen. Okay? That's what happens. Okay. So now you are going to put this stretch receptor there. So you are going to put signal from the stretch receptor to excitatory cell, which will try to create contraction. Okay? So if that happens, this line has shifted. As you can see, this intersection point has moved. Okay? So that shifted, and therefore the so-called equilibrium point is that red dot at a new location. But now notice these arrows. These arrows are essentially vectors created by this equation for each of those points based on this E dot and I dot. Okay? So those are those vectors. So now this point here is not stable. So if it gets disturbed, it goes into this circular, it gets stuck in that circular loop, which is called a limit cycle, which has a period. Okay? So that is what it is. And so what it will happen is you have a single oscillator at each level, which is independent of each other. They are all activated by stretch at the same time by the flip bag. All of them start firing in a periodic way in that limit cycle, and this is how your graph is going to look. Okay? All of them vertical lines because they are getting activated at the same time. Okay? So, and this activation has a period. Okay? So this is a single oscillator period. Okay? So let's remember that. Now imagine this same set of oscillators connected in full circle. Okay? So they are in full circle, So they are being affected by their neighbor. But because they are in full circle, each one looks like their neighbor. Okay? So in fact, you can club these together. So this A plus B becomes, because EI plus 1 is exactly like EI, A plus B becomes that effective A, and E plus D becomes some effective E. Okay? So it still looks like a single oscillator, but with a modified parameter. Okay? So it will also have a same kind of solution with a different period. Okay? So there are these two situations. All right. So I have that, and I can plot a graph. This, of course, this period of oscillation depends on this activation. Okay? How much you have stretched the bag. Okay? And there are these situations here. So now let us look at that explanation for that antigrade contraction. Okay? So what ends up happening is, when you stretch the bag, all the receptors get stretched and activated at the same time. They behave in the beginning, right, like a fully periodic system, because they are exactly like each other, all are getting activated at the same time except for the guy at the very top. Okay? Because that guy is not getting signal from anybody above it. Okay? So first guy is special, otherwise everybody starts firing at the periodic frequency, okay, initially. But then the first guy is going at its single oscillator frequency, which is smaller. Okay? The time period is smaller. Okay? And that is forcing the guys below, because it's a forced oscillation. The top guy affects the next guy, and there is a front of this period correction that propagates. Okay? And that propagation front is basically this white line. Below that, you will see the period is of the periodic oscillator. Above that, it is that of a single oscillator. And while this adjustment happens, there is this phase shift that in which it gets locked, and you end up getting these slanted looking solutions, which are essentially the repetitive antigrade contraction. So basically, we do have a neural circuit which reproduces the behavior that people were challenging. Right? Similarly, you can explain some contractions in the other direction, which I won't get into in the interest of time. So I'm going to skip this. So that issue has been solved, and this now becomes important. So now I have two devices which are capturing two different aspects of the esophagus, and now I can have a disease classification scheme based on combining these two things. Okay? And that part we have done, which I won't discuss here. Okay? So this is that one thing. And then one last thing I want to mention. Okay? I have very little time, but I'm very tempted to do it. Okay? So aortic aneurysm. Okay? So, and this I'm doing just to point out to you, I've talked about non-dimensional parameter as a physiomarker, right? But I haven't shown you a real example of that yet, okay? Although those other ones are just unfolded as physical parameters, okay? So aortic aneurysm, you probably have heard of this, that many times you hear that there is sudden death of somebody because it goes unnoticed, okay? This is the ascending aorta, it bulges, and it gets detected only after it has bulged. Okay? So there is no predictive scheme to actually predict that this is going to grow. All right? So one of the things that we did, mechanics based, was to make this hypothesis like a flag flutter hypothesis. Okay? So if you have a stiff flag and you pass wind on it, it is not going to flutter. But if you make a compliant flag, it will flutter. Okay? So if you use that kind of a basic idea, the whole idea is, as through this aorta, if the aortic wall becomes more compliant, they might tend to flutter, and that is an indicator of future growth of that tube. Okay? So that was the hypothesis that we made, and basically we started with mass, momentum, and tube law equations similar to the ones before, and we did some Floquet analysis. It is not important. I just put these equations to convince you that we did it, but this is going to be out in Nature BME, and what ends up happening is you get a non-dimensional number. Okay, new number, which characterizes that instability. And this non-dimensional number is measurable from MRI data. All right? And what does it involve? Blood pressure, the size, the frequency, kinematic viscosity of blood, and stiffness of the walls. Okay? All of these things are measurable, and this number, if it is large, it is going to create flutter. If it is small, it won't create flutter, and theoretically we can find the threshold. Okay? So we came up with that, and we decided to check with retrospective data. So we took data at time 0 and calculated this parameter for patients and predicted whether it is going to grow or not. And then in follow-up, we saw what the doctors found in the follow-up meeting and whether it had grown or not, okay, consistent with our prediction. And so I'm going to skip that. And this is that result right here, okay. So as you can see, we had basically no false positive and only one false negative over here, okay, in our prediction. So it is working pretty well. So we are trying to do prospective studies to show, and this is an example where we have a non-dimensional number, right, like a disease vector state in the mechanical space, which is a very strong indicator of whether flutter and future growth will happen. And this is extremely important because this is predictive. You might have a subject who has a normal size aorta, but we are predicting this will go bad in the future, whereas current therapeutics, you see it and that person will be considered normal, okay. And this is what I mean by being near a cliff, okay. So this is that example of that. And in fact, we have gone further and shown in experiments. So this is the same thing, but in a non-growing, non-biological thing. This is like an aneurysm. It is in that unstable state and it ruptured, okay. So this is like aortic aneurysm rupture, but in a artificial system, okay. And it is consistent with our theory. So with that, I will actually end. Thank you.
Neelesh, thanks for such a wonderful talk, very enlightening. We have time for some questions. Yes, George.
Yeah, thanks. I mean, I enjoyed all of your talk, and I have about a million questions, but the model with the coupled oscillators couldn't help reminding me the famous paper by Alan Turing in 1952 on the mathematical basis of morphogenesis, where he basically showed that if you couple oscillators on a ring, you can get pattern formation. And then, of course, this is the origin of reaction diffusion equations and so on. Can I think of this in the same framework if I go to a continuum, for example? Or am I like totally off base here?
No, that you're on the right track. The only difference is that in this case, I use that circled example just as a solution that starts at the beginning, but in the end, it becomes a forced oscillation problem. So the first oscillator is basically deciding what happens. So it is not the ring solution in the end. Yeah, but otherwise you are thinking on the right pattern. In fact, that is how we started thinking about it.
And of course, you're doing parameter estimation, right? So you're trying to estimate the coupling.
Exactly, yeah. And there is a rich parameter space, which obviously I simplified everything for pedagogical reasons, but there is this whole phase diagram and everything that we have. And it gives rise to fairly, I'll give you an example. So you have a bag, you inflate it too much. In certain situations, if you inflate it too much, it will cause certain behavior, and if you don't inflate it enough, it will cause that same behavior. So there is that middle part where it is normal, below it is abnormal, above it is, and so obviously all that I didn't talk, but you are on the right track. Yeah.
Other questions? Neelesh, in terms of the esophagus, do you have, like if we change the boundary condition to a slip boundary condition and so on, how do those things affect?
Right, no, that is a good question. So in fact, that blue layer that I showed, which kind of slides, we call it the solid lubricant, which brings in an effective slip for the bolus as it goes down. And if we remove that, making it completely effectively no slip, then you need extremely high pressures to make the bolus go through, okay? So in fact, until that work that we did, the functional role of the submucosal layer was considered to be not any important, okay? It was just occupying space. Functionally, it was not doing anything else. But we showed that actually if it is not there, okay, and if it was stiff, you would find it very hard to swallow, okay? So there is that importance of slip, yeah.
We are actually beyond time today, so
Yeah. Sorry about that, yeah.
It was fun. We'll stop here, and if you have any other questions,
Yeah, I'm here.
please. Thank you very much.
Thank you.